Okay, this is the last lecture on microbial metabolism. Okay, anaerobic pathways. As we learned in the previous lectures, anaerobic means it is a condition where there is a lack of oxygen, right? So in anaerobic pathways, pyruvate from the glucose in the absence of oxygen will take either anaerobic respiration or fermentation. Anaerobic respiration is similar to aerobic respiration, but then the only difference in the final electron acceptor in the ATC. So in anaerobic respiration, the final electron acceptor will be an inorganic molecule other than oxygen, for example, sulfate. And on the other hand, fermentation, it is a process which pyruvate acid converted to a product such as alcohol or organic acid, for example, lactic acid. Okay, there is a video here made by a friend of mine whose name is Dave. So let's watch this video together.
In the biochemistry series, we looked at aerobic respiration. This is the process by which the cells in our body generate ATP through glycolysis, the citric acid cycle, and oxidative phosphorylation. This third step is the one that produces the bulk of the ATP payout, and it relies on the presence of molecular oxygen, because oxygen is the final electron acceptor in the electron transport chain. So without oxygen, this process cannot occur, hence the term aerobic, which means involving oxygen. But there are other methods of ATP production that do not rely on the presence of oxygen that we will want to understand now that we have begun to look at different kinds of organisms. Those would be anaerobic respiration and fermentation. The latter is used by animals such as ourselves when the body is not getting enough oxygen to fuel strenuous activity. And both processes are also used as a metabolic pathway by certain microorganisms that exist primarily in environments without oxygen. So let's get a closer look at these now. With anaerobic respiration, an electron transport chain is still used, but the final electron acceptor at the end of the chain is something other than molecular oxygen. For example, some bacteria will use a sulfate ion and produce hydrogen sulfide instead of water as a byproduct of this activity, which produces an odor of rotten eggs that you may have smelled in certain environments. Fermentation, on the other hand, does not involve respiration of any kind. This will always begin with glycolysis, just as we learned before, as this step does not require oxygen and does result in the production of 2 ATP per glucose molecule. But the pyruvate that results with oxidation by NAD+ will not feed into the citric acid cycle. Instead, something else will happen, depending on which type of fermentation is occurring, so as to regenerate NAD+ from the NADH that is produced, allowing glycolysis to continue. The two types of fermentation are alcohol fermentation and lactic acid fermentation. With the first of these, glucose is oxidized over the 10 steps we learned about to produce 2 pyruvate molecules and 2 ATP molecules, using 2 molecules of NAD+ in the process. Then each pyruvate will undergo decarboxylation to produce acetaldehyde. And finally, acetaldehyde is reduced by NADH to produce ethanol. This allows for the regeneration of NAD+, which can then be used in glycolysis again. The final product, ethanol, is the kind of alcohol that humans consume, hence the term alcohol fermentation. And this process has been utilized for centuries to produce alcoholic beverages such as beer and wine. Yeast, which is a unicellular member of the fungi kingdom, performs alcohol fermentation. So this organism is used in these processes to produce the alcohol we drink, and also by bakers, where the carbon dioxide gas produced during the decarboxylation step is what causes bread to rise. Next, looking at lactic acid fermentation, again glycolysis occurs precisely as we have learned, but this time the pyruvate that is produced will not decarboxylate. Instead, it will be directly reduced by NADH to produce lactate. This again allows for the regeneration of NAD+ for use during glycolysis, and also involves no release of carbon dioxide. Lactate is the conjugate base of lactic acid, and it is the production of this compound by certain fungi and bacteria that allows them to be used in the industrial production of cheese and yogurt. In addition, human muscle cells will resort to this process when the oxygen supply is running low, most typically during prolonged periods of strenuous exercise, when glucose catabolism is going faster than our ability to breathe enough oxygen into the bloodstream to sustain the activity. In such a case, the cells will switch over to fermentation, allowing for a brief burst of additional energy production. But the lactic acid that builds up in the muscles during this activity causes muscle fatigue, which limits the duration that this activity can be sustained. So to summarize, all of the forms of energy production that we have learned, whether aerobic or anaerobic, begin with glycolysis. This is therefore the most evolutionarily ancient method of ATP production and must have evolved in very early forms of prokaryotic life, given that it occurs in the cytosol and not in any membrane-bound organelle, such as mitochondria, which did not exist until eukaryotes came about. This makes sense with what we know about Earth's early atmosphere, which did not contain oxygen until the evolution of photosynthetic cyanobacteria, which prompted large-scale oxygenation of the atmosphere, making the evolution of aerobic respiration possible. In every living organism, once glycolysis is complete, pyruvate may be reduced through lactic acid fermentation or broken down and then reduced through alcohol fermentation. It can also be fed into the citric acid cycle to continue with aerobic respiration or anaerobic respiration if something other than oxygen is available to act as an electron acceptor in the absence of oxygen. In all of these processes, the NADH that forms when NAD+ is reduced during glycolysis must be oxidized to regenerate NAD+, and this is achieved either when NADH reacts with pyruvate or acetaldehyde during fermentation, or when NADH dumps electrons into the electron transport chain during respiration. The main difference is the electron transport chain drives oxidative phosphorylation, which produces most of the ATP generated during cellular respiration, whereas fermentation produces only 2 ATP, given that it is comprised only of glycolysis, so it's a huge difference in terms of energy production.
Okay, thanks Dave. Okay, in summary, anaerobic respiration is similar to aerobic respiration in that electrons are extracted from glucose molecule and passed through an electron transport chain to synthesize the ATP. On the other hand, in fermentation, pyruvic acid is metabolized to alcohol and carbon dioxide in alcoholic fermentation. Okay, we are done with glucose metabolism, so let's start with the lipid metabolism. Okay, lipid catabolism. So the fat will be digested into glycerol and 3 fatty acid by the lipase enzyme. Now after that, this glycerol will be converted into dihydroxyacetane phosphate, which is an intermediate in glycolysis, and this will go into the glycolysis and converted into pyruvates. And on the other hand, fatty acid will be oxidized into acetyl coenzyme A, which is going to be involved in the Krebs cycle. And therefore, the oxidation of glycerol and fatty acid produces intermediates that can be used by glycolysis and Krebs cycle for further oxidation and energy production. Okay, let's briefly see the protein metabolism in bacteria. Okay, next is the protein catabolism. So as we know, protein composed of polypeptides, and then this polypeptides is composed of amino acids. So usually proteins are too large to pass through the plasma membrane of microorganism. Therefore, they need to be digested or break down into its monomer, which is usually using an enzyme called protease, and also protease known as a peptidase or proteinase. And in this proteinase is an extracellular enzyme that breaks down the protein into amino acids. And amino acid then converted into intermediate that can be used in the other metabolic pathway to produce the energy. Okay, let's briefly go through the cell biosynthesis. Okay, part of the energy that produced via catabolism of a carbohydrate, lipid or protein is used to produce new cell component. The main classes of biological molecules synthesized by cells include polysaccharide, lipids and amino acids. Okay, microorganisms synthesize lipids by unifying glycerol and fatty acids. And fatty acids are hydrocarbons and form from successive addition of two carbon fragments of acetyl coenzyme A. And the enzymatic linkages of glycerol to fatty acid results in a biosynthesis of a variety of simple fats or lipids. Okay, let me briefly go through the importance of lipids. So lipid is a structural component in the cell plasma membrane, and it also helps to form the cell wall of gram-negative bacteria. So similar to carbohydrate, lipid also can be a source of energy, and the lipid also form intermediate products after biological oxidation and then feed into the Krebs cycle. Okay, synthesis of amino acids. So as we know, amino acids are building blocks for protein synthesis. Some bacteria like E. coli, they're very smart. They can synthesize all the amino acids that need from glucose and inorganic salt. But there's some bacteria like lactobacilli that were not able to make the amino acids they require. Therefore, they acquire the amino acids by catabolizing the protein, where we provide the protein in the media. And therefore, the glycolysis and Krebs cycle are important sources of precursors needed for amino acid synthesis. So therefore, we are done with the microbial metabolism. So if you have any question, please post in the e-learn and we will discuss tomorrow.